Hello lovelies, in this video the brilliant Dr. Edwards is going to be taking you through our OCR Gateway Biology Paper 1 Separate Science Foundation paper, and there probably aren't many people watching this video, but this is our predicted paper for 2023. So if you haven't had a go at this paper already, what I want you to do is to pause after the question comes up, have a go at it yourself on a bit of paper, play it, and then we can all work through the answers together.
Hi everyone, so we're looking at the OCR Gateway or OCR-A Biology Paper 1, but it's for the separate foundation tier. Hopefully you've downloaded the paper and had a go at it at exam conditions in the time that's suggested on the front, and then maybe you've had a go at marking it already, or maybe you want to mark along with me. That's fine. What we need to do is we're going to go through each question step by step. I'm going to analyse the questions, give some exam tips, so what you should highlight, what you should annotate, link to kind of what content you should know to be able to answer that question, then give some model answers, and then explain the mark schemes. So hopefully this will help you identify whether it was that you didn't know the content for that question, or you didn't necessarily understand what the question was asking you for, or you didn't hit all the mark points in the mark scheme. And hopefully we'll be able to spot those, and then you can use those to help you with your revision. We've got grade boundaries on the front of the paper. They're very rough guesses. So if you want to have a go at it and then work out your percentage, doesn't necessarily guarantee that's the grade you'll get in the real exam, but it's worth having a go and seeing what percentage you get, and kind of tells you roughly where you are. Okay, so have your paper at the ready and off we go. Okay, so we're going to start with multiple choice questions in section A. Try not to spend too long on this section because they are supposed to be quite quick and they will need some working out, but you shouldn't spend really long time working out questions if you do get really stuck. The image below shows dividing root cells as seen with a light microscope. Questions 1 to 2 refer to the image. Which letter in the image represents the cell in the growth stage of the cell cycle? So if you remember the cell cycle, growth stage is not dividing. The cell isn't dividing, so shouldn't be seeing anything that looks like mitosis here. The cell is just growing. It's kind of in interphase. You may have learned it as that, but it's just in a stage where we're not going through mitosis. So you're trying to look for clues in the pictures that would either tell you if it's doing mitosis or not. And mitosis, we can always tell, is happening because chromosomes are moving. So if you can see chromosomes and you can see chromosomes moving, being pulled apart, or meeting in the middle, or moving around in the cell, then it's going through mitosis. And it's going through cell division. It's not just sitting happily and getting bigger and growing. So D is clearly dividing, C also clearly dividing, and B is clearly dividing because we can see chromosomes and we can see them moving around. Whereas A, there's no chromosomes moving, you can't see them moving around, we've just got a nucleus with the DNA in it as normal. So there we go, we are in growth phase. So our first answer is A. Question 2. There are 42 whole cells in this field of view. What percentage of the cells are undergoing mitosis? Okay, so we've already identified that B, C, and D are dividing because there's chromosomes moving. So they're undergoing mitosis because they're in various stages of mitosis because the chromosomes are either being pulled apart or they're in the middle. So therefore we know there's 3 cells that are undergoing division out of the 42 whole cells. There aren't any other cells dividing, you can just check that none of the others were labelled, but there are no other cells that look like that. So it's 3 out of 42 times 100 to give us a percentage, and our percentage comes out about 7.14, which means it is C. If you ever do calculations in these and you get very, very close to one of the answers, I would say if you're not 100% sure, then that's probably likely the answer to go for. It is likely you could have done something wrong with the calculation, but if you get very close but not quite, it could be a rounding issue. I would say go with that answer if you're not sure. Remember, for these, you always need to put something. You should never leave a multiple choice question blank. Question 3 then, what's the maximum resolution of a transmission electron microscope, otherwise known as a TEM? This one got a very high resolution, the highest resolution of all the microscopes. It can actually see down to individual atoms. It's been used to visualize an atom. So we need the smallest number. This is one of those numbers you can just learn. So you can just learn that the resolution of an electron or TEM is 0.2 nanometers. But on this list, if we weren't sure, I'd always go with the smallest for a TEM because it really, really is very, very small. Okay, so on to question 4. The figure below shows a plant cell and its organelles. Which letters correctly show the 2 organelles involved in protein synthesis? Okay, so we're picking 2 letters. We've got only 2 letters to choose from. So we need 2 organelles that are involved in the process of protein synthesis. Protein synthesis is where we take DNA, we turn it into mRNA, and then we turn that mRNA into a chain of amino acids or a protein. So we've just got to think about where in the cell do the stages of this happen. So the DNA and mRNA occur in the nucleus. So we make the mRNA in the nucleus, and obviously then it leaves and it goes to the ribosome, which is where we make the protein. So our 2 organelles involved are the nucleus and the ribosome. So hopefully we can identify S as the nucleus because it's the big round circle structure in the middle. And then we need to look for a ribosome. So we might think that it could be one of these dots that's labelled just loose in the cytoplasm. But most of the time that's just to represent cytoplasm. And none of those labels are pointing to any of those dots anyway. Other location where ribosomes can be is in the rough endoplasmic reticulum, or the RER, and that is labelled N. You can see the dots on there. So that is another place where ribosomes are located. And so then clearly our answer must be D, because it's where the answer S and N are the letters to represent those 2 organelles. You could do this by process of elimination. So if you look at S and M, that would be the nucleus and then a structure you're maybe not sure about what that is. So I would say unlikely. S and Q, so still the nucleus. But if you look at Q, that is again a structure that you're not sure about. And then N and O obviously doesn't involve S, so it can't be that. So lastly, it's S and N, and that makes more sense. Okay, so on to question 5. Which cellular transport processes require ATP? This is the same as asking which transport processes require energy, because that's what ATP is. Hopefully we know that diffusion and osmosis are both passive forms of cell transport. So they do not require energy. Only active transport requires energy. That's why it's active, because we're going against the concentration gradient. Okay, on to question 6. What biological molecule does the following statement describe? Double-chain polymer with sugars joined to phosphate molecules. Okay, this might be quite easy and obvious, but let's work through the answers just in case. So protein and carbohydrates are not double-stranded. Are polymers, because they are made up of obviously repeating elements, but they're not double chains. Proteins are made of amino acids, and anyway, not sugars and phosphate molecules. Carbohydrates are made of sugars, but they're not, doesn't include phosphate molecules. Carbohydrates only contain carbon, hydrogen, and oxygen, hence their name. So RNA, it could be, because we should know that nucleic acids or DNA and RNA are polymers. They have sugars and phosphate molecules, but RNA is only single-stranded. So that means the answer must be DNA, if you didn't get that already. Which of the following statements about translocation is correct? So let's double check that we're happy with translocation. So we need to see which of these statements about the movement of sugars in the phloem of plants is correct. This is what I would write, maybe even brain dump a bit more about what you know about sugar transport or translocation and movement of sugars in the phloem. First of all, food substances move down the xylem from shoots to roots. Well, that involves the xylem, so it can't be that. We know we need to talk about the phloem. Food substances can move up and down inside the phloem tubes. Yeah, that's correct. Let's check the others. Food substances and water travel up from roots to shoots. Well, food substances, but we know that sugars and things are made in leaves, so it can't be about translocation then, it's about water travelling. And then food substances travel downwards from the shoots to roots in the phloem. It could be, but we know that actually they don't just move down, they move up and down. So it has to be the second one, which is B, because that is talking about bidirectional movement or up and down movement inside the phloem. That makes sense, that is correct. Okay, on to question 8. Which of these statements describes negative feedback? So negative feedback is where we're always trying to return something to the optimum, or maybe you could say reverse a change that is moving away from the optimum back towards the optimum. It's returning conditions to normal if they move away from those conditions. It could be up or down, and the change could therefore be up or down depending on whether we're trying to bring something that's gone too high back down or too low back up. So when the body responds to a stimulus by increasing the change, not necessarily always just that, so can't have A. When the body responds to a stimulus by changing the internal environment, Could be. Doesn't say how we're changing the environment though. So I would say it's not necessarily specific enough as an answer. When the body responds to a stimulus by reversing the change, yeah. So if it went up, we would bring it back down. That's reversed. If it would go down, we'd bring it back up. That would be reversed. So I think that's fine. Let's check D. A continuous cycle of change in the internal environment. Negative feedback loops are pretty continuous, but they're not always happening. It depends on the stimulus and whether the external environment is changing as well as or the stimulus is changing. So it's not always going to be a continuous cycle based around the internal environment. So it is C, which is where we take a stimulus, we respond to a stimulus, and it reverses the change. Question 9. The graph shows an enzyme-controlled reaction carried out at optimum temperature. So we've got rate of reaction on the Y and enzyme concentration on the X-axis. And it's asking us what is the limiting factor at part B. So hopefully we've seen graphs like this before. It doesn't really matter what the reaction is. These graphs will always look the same, really. So we need to think about what's happening at A versus what's happening at B. So at A, the limiting factor is the enzyme concentration, because as enzyme concentration is increasing, the rate of reaction is increasing. That means at B, it must be something else. It can't be temperature though, because if we think about all the things that can affect the rate of an enzyme-controlled reaction, temperature is the one we think of maybe first. But in the question, it said this reaction is being carried out at the optimum temperature. So it can't be that. So then what else can affect enzyme rates of reaction? Well, pH is one of them, or the substrate concentration. So how much substrate is there for the enzyme to actually work on? Once that runs out, then the rate will stop, or the rate will maintain until all of the substrate has been reacted with. So not enough enzyme, nope. Temperature, we've said it can't be that, nope. Not enough substrate, there we go. Too much product, that's not going to affect an enzyme reaction. So we get C. This is what I mean about trying to make sure you've annotated as much as possible. Brain dump, write down lists of things, lists of bits of content that you know link to this question. Because then it makes it easier, because then when you look at the multiple choice answers, you already ruled some out. And if one of the other answers isn't there, this made this a very easy answer to get. Okay, so on to question 10. In which part of the heart is the heartbeat initiated? Okay, so this is one of those things that we just have to know. So the heartbeat is initiated by a group of cells called the pacemaker cells, and we just have to know that these pacemaker cells are found in the right atrium. Which two hormones are produced by the pituitary gland? You need to select two letters, so we're not just putting one letter this time. So the pituitary gland is found in the brain. Hopefully we know that estrogen and progesterone are produced by the ovaries, so that leaves us the other two, which is LH, luteinizing hormone, and FSH, follicle stimulating hormone. So our answer is A and B. 12 then, if 30% of the bases in a piece of DNA are T, what percentage of the bases are C? Okay, so this is just a maths question, but it does rely on us knowing that all T bases will be bonded to A bases. So if 30% of the bases in a piece of DNA are T, there will be exactly the same amount A, because they are bonded together, so it has to be the same. So that means that accounts for 60%. Two lots of 30% gives us 60%. So we need to work out what's left. So 100% take away 60% gives us 40%. And then the same applies for C and G. So there's got to be the same amount of C as there is the same amount of G bases, because they are bonded together. So we just need to split that 40% into two. So two lots of 20%. So that means that the percentage of C bases are going to be 20%, and that gives our answer as C. Question 13. The use of electrons as a source of radiation in the electron microscope allows high resolution to be achieved because electrons are negatively charged, have a very short wavelength, can be focused using electromagnets, or travel at the speed of light. So the first three statements here are true. They are negatively charged, they do have a short wavelength, and they can be focused using electromagnets. They do not travel at the speed of light. That's false. So we don't want D. We know it's not D anyway. The focus of the question, though, is asking us why or how this fact about electrons can help us get high resolution. So this is knowing and understanding what resolution is. So it's seeing 2 points that are close together as separate objects. So the fact that electrons are negatively charged is not going to have anything to do with this. And obviously they can be focused using electromagnets, but focusing, remember, gives us a clear image, a non-blurry image. The seeing two objects as separate objects is due to a wavelength. If I have 2 points close together in a large wavelength, you'll see the wave just passes over the second point. If I have a short wavelength, the wave will hit both points, which allows us to see them as separate objects. So our answer is B. Okay, so question 14. Which of these structures is only found in prokaryotic cells? The only is in bold here because it's important. It's got to be something from this list that is only found in bacterial cells. So that's what prokaryotic cells means if you weren't sure. Prokaryotes are bacteria. Eukaryotes are animal and plant cells. So it's got to be something from this list that only bacterial cells have and that animal and plant cells do not have. Okay, so we've got ribosomes. Well, no, we know animal and plant cells both have those. You've got a cell wall. We know that plant cells have a cell wall, although prokaryotic cells have them as well, but we can't be the right answer because plant cells have it too. Plasmids. So these are the loops of DNA that are found in bacterial cells. So we know it could be that. So let's just check. The last one is nucleus. Well, we know that prokaryotic cells don't have a nucleus because that's what separates them from animal and plant cells. So it must be plasmids. So our answer is C. Okay, so on to our last multiple choice question, number 15. The concentration of blood glucose, as well as the concentrations of the hormones responsible for glucose regulation, were measured in the blood in the hours after a high carbohydrate meal. The graph shows the results of the test. So they've eaten a high carbohydrate meal, so a meal that's going to have lots of glucose in it when it gets broken down at time 0, and then these are the hours after that's been eaten. It wants us to identify which letter shows the correct labels for Z and Y. So we've got blood glucose labelled on the graph. So that must mean that the other two are the two hormones that control glucose regulation. We can see that the black line Y increases as blood sugar levels increase, and the dotted line for Z decreases as blood sugar levels increase. So that should tell you a bit about the relationship between the hormones and what they're doing. So which hormone that controls blood glucose? There are only two. We've got insulin and glucagon. Which one is going to increase when blood sugar increases? So which hormone will be released when blood glucose levels are high? So this relies on us knowing that insulin lowers blood glucose and glucagon raises blood glucose levels. I would always like to remember it as saying, when the glucose is gone, you need glucagon, or you release glucagon. Glucose gone, glucagon. So when there's not enough glucose in the blood, you release glucagon. That's how you remember that insulin does the opposite, so it lowers blood glucose levels. So it's going to need to bring blood glucose levels down. And you can see that after we get this peak in Y, then the blood glucose levels do start to decrease, and then so does Y. So pretty happy that Y is insulin, which means that Z must be glucagon, because that is the other hormone involved in regulating blood glucose. So now we just have to pick the right answer from our list. So it's not C because that's the opposite way around. It's not D because that doesn't have both the hormones in it. And so we just check it is B because we've got glucagon being Z and insulin being Y. Okay, so that's our end of our multiple choice section. Okay, so on to section B and our first question. These are now obviously longer answer questions. To look for the presence of starch in potatoes, a student decided to observe potato cells under a microscope. Describe how the student could have used the equipment above to prepare a slide to view under the light microscope. Okay, so we've got some pictures of equipment. Sometimes you'll see questions like this where they want you to describe how to do something, which just means like method steps. So it's probably going to be related to a practical that you should have done before or seen before. And so hopefully with this one, we're kind of thinking, okay, I've seen this equipment before, and I'm going to have to mention it in the steps. So when I talk about the steps, they need to be just like detailed method steps about how to use all these bits of equipment. So I've got to name them. If we're using a light microscope, that's important because we need to remember that our pieces of tissue need to be thin. So think about practicals that you've done that are like this. So like if you've looked at onion cells or if you've looked at cheek cells. This is more like onion cells because we're looking at plant tissue. But the idea that we need a thin piece of tissue to lay on a slide so that we can let the light go through it, and we might need to stain it as well. So I've got my potato on some kind of board or a tile. I've got my knife or scalpel. I've got my microscope slide. I've got a smaller glass slide, so this will be the cover slip. And then I've got a pipette, which must contain some kind of liquid. And so, like we said, we could put stain on this slide. We could put a drop of water on this slide. If you're going to say stain and be specific, let's think about what this question is asking us. So we're looking at the presence of starch in the potato cells. So what stain could we use? Well, similar to the onion cells, we can use iodine because that would show up starch. That's a test for starch. We're going to probably need to use that as well in the next question. So this is just what step by step am I going to do to cut a thin piece of tissue of my potato, put it on my microscope slide, put a drop of stain or water on it using a pipette, and then cover it with a cover slip. Remember, with a cover slip, you do have to lay it down gently at an angle in order for us not to get any air bubbles under there, because we don't want to see air bubbles, we would like to see our actual cells. So that's all I'm going to do. I'm just going to write these out in 3 steps because it's 3 marks, so that makes sense. So cut a thin slice of potato tissue and lay it on the microscope slide. Make sure to say the word thin. Use the pipette to place a drop of water or stain onto the tissue. And then gently lower the cover slip at an angle to avoid air bubbles. Okay, so the next part of the question says, the image below shows starch grains present in potato cells viewed at a magnification of ×400. What test could the student have used and what result would be seen to show that these grains contained starch? Okay, well, we've already talked about it. We've already talked about the fact that we could use iodine, and then we need to say what will happen when we put it on these cells when it comes into contact with the starch that shows us that these grains contain starch. So what's the positive result if you add iodine to starch? Well, hopefully we know it goes blue-black. There's a clue in the image as well, though, because it shows you that you've got some starch grains in the image and they are this kind of blue-black or black colour. So there's a clue if you weren't sure, but you should hopefully know the result, positive result for iodine with starch is blue-black. So I'm just going to say what we're going to do. So add iodine to the tissue and the starch grains will turn blue-black. Okay, so on to part C. Calculate the actual length in micrometers of the starch grain between points A and B. Okay, so we sort of expected this maths question when they gave us the magnification in the first part of the question. We're looking for actual size, so we're going to use our magnification, image size, actual size equation. Careful, they want it in micrometers. So we need to convert our units at some point. We need to convert from what we measure in to our final one.
So just be careful and make sure you don't forget that step. So the first thing we need to do is actually to measure our line.
So I've given you a little picture of my ruler here. This is what I would do. Always have a see-through ruler. It's so much easier. And you need to measure it accurately. So make sure you double check and do it a couple of times. And we're measuring in millimeters, not centimeters, because then you're going to get yourself in a pickle because it's harder to convert from centimeters to micrometers than it is from millimeters to micrometers. We're going to be using our equation, which I always remember as I am a triangle. Put it in a triangle, and then you're always going to be able to remember which way round your equation needs to be. So we're working out actual length or actual size, so that we're going to do our image size divided by our magnification. We've got our magnification in the question, it's ×400. So we just need to measure our image in millimeters in order to get the image size, and then we're good to do our calculation. Here you can see image size is 14 millimeters. We need to write out what equation we're doing. So actual size equals image size divided by magnification. It's always worth doing this and writing out your equation once you've rearranged it, because you can get marks for this. You could even write it out before you rearrange it, and again, you may still get marked just for writing out the equation you're going to use. So our actual size equals 14 millimeters divided by 400, which is our magnification. Remember, magnification doesn't actually have units, so we don't have to worry about that too much. Then we need to convert to micrometers before we do our division. I would always say do this so that we don't get ourselves in a muddle. To convert from millimeters to micrometers, we just times by 1,000. so it's 14,000 micrometers. Do that calculation on your calculator. You get a calculator for a reason. Don't try and do anything like that in your head. And so we get an answer of 35 micrometers, which sounds about right if we think about the fact that obviously we looked at that image with the ruler and we've measured that, and we know this has actually been blown up 400 times. So that makes sort of it's a good estimate. Part D then, the last part of this question. Plants store excess glucose as starch in their cells because it's insoluble. Explain another way plants use glucose in the cell. So the knowledge this question is testing is our uses of glucose in plants. So we should have a few examples in both plant and animal cells of how they use glucose. But in this case, we need to give reasons for what that glucose is used for. So not just say, glucose is used, for example, in respiration. Yes, but how is it used, or why is it used in respiration? Well, to release energy for the cell. One that's good for plant cells for this example is thinking about the fact that glucose can be used to make cellulose. Again, just saying used to make cellulose isn't enough because we need an explanation. So what's the cellulose used for? It's used to make cell walls. So we can choose either of those. And there's some others as well we'll look at in the mark scheme. And then we're just going to use that as our answer. So it's used in respiration to release energy for the cell. Okay, so this is the mark scheme for question 16. Lots of options for the 16A, so a thin slice or a thin section. Put on slide in water, solution, stain, iodine, any of those. And then you've got your lowering your cover slip. Add iodine to the slide, or cells, or tissue. And then you've got turns the grains blue, black, or black. You can't just say blue. You can't say purple. Mark schemes with the iodine test results are always going to accept blue, black, or black, but that's pretty much your only options. If you've said anything else, you can't have that second mark for 16B. Use measured length between points A and B and uses magnification are the two kind of steps you need for 16C to get the 2 marks, and ideally you'd get the correct answer. I've got the correct answer as 35 micrometers, but it's going to depend on your printing. So how you're measuring it. If you didn't manage to print it out, or if you printed it out slightly differently, there's always a chance it could change size. So if you worked out, if you measured it correctly, however you've measured it, so sub 14 millimeters for whatever you measured, as long as you did the steps that I did, you can get 2 marks for your correct answer. But if you didn't get a correct answer, or you didn't do the method steps that I did, as long as you've done the conversion of millimeters to micrometers, then you can have 1 mark. For 16D, main thing here, we talked about the cellulose and we talked about the respiration. You can also say to combine with other elements or nitrogen specifically to make amino acids. Or you could maybe talk about combining to make fatty acids as well. But the main thing here is if you're going to do the respiration answer that we did, you need to say to release energy. You can't say make energy. You can't say produce energy. You could say provide energy for the cell, that's fine. You could say produce ATP instead of releases energy, but as long as you've made sure you haven't said the idea of making or producing, because energy cannot be created or destroyed, rule of thermodynamics, so we can't have that as an answer whenever we're talking about energy in the cell. Okay, so question 17. So the human nervous system is divided into 2 main parts, autonomic and somatic nervous systems. One of the functions of the autonomic nervous system is to control reflex responses to danger. So there really is the bit that this question is actually about. It's about reflex responses to danger. So regardless of whether you understood all of the question, the idea that we should understand or recognize reflex responses is important here. Put the following in the correct order to show a chain of nervous responses, including the name of the neurons which result in a hand being pulled away from a sharp pain. Okay, so we've got some letters here, A to E, with statements, and it wants us to put these in the right order. So put the letters in the right order, those statements would then describe the pathway or the chain of a nervous response. It's basically just describing a reflex arc. So although this one doesn't involve a hand moving, it's the same kind of idea in my diagram. I've got my leg moving when it gets hit as a knee-jerk reflex. So we need to think about what statements we've got and what we know or remember about the reflex arc. So this kind of like round shape here is supposed to represent my central nervous system or my spinal cord. In my spinal cord, we should know that there is the relay neuron, which runs between the other 2 neurons in our sequence. So that's in the middle here. And then I have my sensory neuron joining onto that. So my sensory neuron is attached to the sense receptor. That's why it's called the sensory neuron. So you have sense receptors in all of your sense organs, and in this case we also have stretch receptors in the muscle. But anything that detects the stimulus is a sense organ or has receptors, and that's attached to the sensory neuron, which is then attached to the relay neuron, and then that's attached to the motor neuron, and the motor neuron is attached to the effector. In this case, it's a muscle which causes movement. Think about motor, like a motor in a car causes movement. So it's attached to the muscle because the muscle moves. All of the M's go together: motor, muscle, movement. So then let's think about the order. So obviously I've got my stimulus being detected by my receptors in my sense organs. So then that travels down the sensory neuron to the relay neuron, which is in the spinal cord or the central nervous system. Then that goes to the motor neuron, which then joins onto the effector, which is the muscle that causes movement. So our stimulus was at point 1, so that's going to be B. Then we have it connected to our sensory neuron, so that is D. Cross these off as you go so you don't accidentally make a mistake with the wrong letter. A is then my relay neuron, because we have our relay neuron in the spinal cord. Then we're going to the motor neuron, which is E, and then we're going to our effector, which is in our muscle. And so that is C. And there we go. Suggest how a reflex response, such as pulling a hand back from a source of pain, might be an evolutionary advantage to a species. So why are reflexes or reflex actions important for survival? So because an evolutionary advantage allows you to survive and therefore reproduce. So we need to be able to say that they are fast, because that's why they're important. They're very rapid or they're very fast, and that allows protection from harm or damage. So it protects the body from harm or damage when it happens. And that means that they're more likely to survive, less likely to suffer injury or death. And so you're more likely to survive and reproduce. And therefore, that's why it's evolutionary advantage to be able to survive these things. Okay, give 2 other examples of autonomic responses by the autonomic nervous system. So apart from pulling your hand away from pain, so not a pain response, give 2 other reflex responses. So these are things that happen automatically without you thinking about them, that can protect you from danger. They're not things like jumping out of the way of a car or being able to catch a ball if it's thrown at your face. Those you have to think about. Your brain has to process information in order for you to react in those situations. We're talking about kind of reflex reactions that are natural, you're born with them, and they don't involve the conscious brain. So things like blinking if something comes into your eye, a knee-jerk reaction when someone hits your leg and your knee kind of bends, pupil reactions, so contracting or dilating of your pupils in response to light, sneezing if something like pepper or dust or something goes up your nose. All of those are something that happen without you thinking about them, that happen quite quickly. So I'm going to go for when your pupils contract in bright light, because that protects you from harm, and when your knee jerks when your leg gets hit. Okay, on to part D. Investigating the nervous system has proved extremely difficult for medical science, especially in the area of the brain. Give 2 reasons why investigating brain activity is considered extremely difficult. So we need 2 reasons for 2 marks. The main thing here is it's hopefully a list of things that you've kind of looked at or discussed about why it's hard to investigate the brain at all. And it's partly because in order to investigate brain activity, you need to be able to compare normal brain activity with brain activity that's broken or faulty. So you need to be able to compare normal brains and damaged brains. Also, to do this, you need volunteers. So first of all, you need to be able to find people who have got brain damage or some kind of difference to their brain activity, and they can be quite rare. And then also you need to be able to have them to be willing to take part in medical research. And they have to volunteer and they have to give up their time to do it and agree to do it, because otherwise then there's an ethical sort of issue there with not having consent to be able to be researched upon. And we need to have that in order for this science to be valid and in order for it to be ethical. There's kind of 2 points there. There's some others and we'll look at them in the mark scheme, but they kind of stem around the same ideas here, that it's rare or it's hard to find patients with brain damage because they're quite rare, and that are willing and able or want to volunteer for this research.
What method
What method can be used by doctors to check if the brain has suffered damage? So this is like an investigative method. How can we look at the brain to see if it's had some damage in a relatively safe way? Well, it will be a scan in order for us to be able to see and visualize the brain inside the skull. And the normal brain scan that we use is an MRI. We'll look at some other examples of answers to this question in the mark scheme, but I'm going to go for MRI.
Two students wanted to compare the function of their nervous system by testing their reaction time using a ruler drop test. Hopefully we've recognised this already as the required practical for paper 2. There aren't many required practicals for paper 2, so it's definitely worth learning the few that there are. So this is the one where we're like using ruler drops to test our reaction time. So they used the following method. Student A held the ruler just above student B's hand. It doesn't say which hand, so that's kind of important, and we'll talk about that as one of the variables in a minute. Student A let go of the ruler without warning, and student B had to catch it with their thumb as soon as possible. The reading on the ruler where the student's thumb was in centimetres was recorded to show how far the ruler fell before it was caught. They repeated this 5 more times, then swapped over to student A was catching. So they are measuring the number of centimetres that the ruler fell through their hand before they caught it. And they did repeat the experiment, which is good. It's important to repeat experiments, we should know, and it means that we've got some sort of reliable results. Okay, so the actual question E says, state one factor the students could have controlled to make this investigation fair. Well, it's basically just asking what should be kept the same for both students. And so anything that should be controlled is something that can affect what you're measuring. So in this case, we need to think about all the things that can affect reaction time, and they're all the things that should be kept the same unless you are changing them. So they're not testing to see if there's any specific effect of tiredness or caffeine or anything in this practical. They're just looking to see what the difference is between the two students. So we need to think of things that can affect reaction time. So how tired you are, your age, your sex, how much practice you've had, how much caffeine you've had, whether you've used the same hand. So in this case, that's kind of linked to practice, but if you use a dominant hand, that's going to be stronger than a non-dominant hand, and you're used to catching things, so you'd expect your reaction time to maybe be faster with your dominant hand. So all of those can affect reaction time. I'm going to say the students should both use their non-dominant hand, but you could say they both need to be the same age, they both need to be the same sex, they both have to have not practiced before, so they're using their non-dominant hand, or they both had to make sure they hadn't had the same different amounts of caffeine that day. One had a coffee and one didn't, and that wouldn't be fair. So any of those that can affect reaction time we can use as a control factor.
Okay, so now we can see the results. After converting their ruler measurements to reaction times using a table, hopefully you remember doing this, so obviously the actual distance isn't your result, you need to convert that to the reaction time in seconds. So they've got the following results. They want us to calculate the mean reaction time for student A. Now this is a 2-mark answer. So first of all, how do we calculate the mean? Well, we add up all the numbers and divide by the number of values. However, if you're calculating mean, you should know that you should do that, but only do that once you've removed any anomalies. So we need to take out any anomalous results and then add up all the numbers and divide them by the number of values. So I'm looking at the list for student A, so we've got 0.27, 0.4, 0.23, 0.24, and 0.22. Now the 0.4 is jumping out at me immediately as a potential anomaly because it's nearly double all of the other values. And if you look over to student B as well, their highest was 0.31. So it's way higher than any number on the table, whether it's for student A or student B. So I would say that this is most likely an anomaly, given how close in range all of the other numbers are. They're all in the 20s. I would say we remove that, and so then we calculate 0.27 plus 0.23 plus 0.24 plus 0.22. That gives us a total of 0.96. And then we're dividing it by 4 because we only had 4 values, so that gives us 0.24. Okay, part H. Give one conclusion you can make from these results. If you're looking for conclusions, especially in the table, then you need to look for patterns or differences that you can talk about. So there's a pattern that I see, which is that for both students, the reaction time decreases as they have more goes. So it decreases with practice, which might be something that you looked at when you did your reaction time. And also we have a difference. So student A's mean reaction time is lower than student B, so they have a faster reaction time than student B. So there's a difference and there's a pattern. I'm going to go for the difference because that's kind of what they were investigating, was they were comparing their reaction time.
Okay, so here's the mark scheme for question 17. For question A, if you got any of those letters in pairs in the correct order, you can have 1 mark if you got that order wrong. You can have 2 marks if you got any 3 letters in the correct order. So if you got BDA, DAE, or AEC anywhere in those boxes in that order, you can have 2 marks, and you get 3 marks if you got them all correct. So obviously if you got them all correct, you get 3 marks, and then you only need the other instructions if you didn't get them all right. For B, you've got fast or rapid for the first mark, and then the second mark is to reduce risk of damage or harm, or to increase chance of survival. So that's how it's linked to evolution. You need that part really to sort of talk about the fact that it's an evolutionary advantage because you're going to be able to survive and therefore reproduce. And that's how obviously we talk about evolution happening. You've got any 2 from this list for part C. So it must be one of these 4 options, or sorry, it must be 2 of these 4 options, because they're true reflex actions. You can't just say flinching if someone throws a ball at you, or jumping out the way of something dangerous. Like, that's not a true reflex action. So it's got to be from this list. And then lastly, you've got 2 from this list about talking about why examining the brain is really hard. So as we said, people who live with brain damage injuries are rare, or people who are, who have these brain damage injuries are rare because they often have a reduced life expectancy. So that's kind of the first 2 are how they are linked. People are not always willing to take part in the medical research. That's in general, like volunteering for medical research is quite a rare thing. You're not always going to find people who are going to just consent to have whatever done. And then you've got ethical issues with patients not being able to consent. So if they're not able to be aware of what they're consenting to, or able to give consent, and it would be coming from a carer or someone who is responsible for them, that's obviously not necessarily an ethical way of doing it. So you've got kind of the links between there being people willing to volunteer, or the consent being given, versus how rare it is to find people who have the brain damage, or people who are, because their life expectancy is often shorter. There's lots of options for the next part. So you can have an MRI scan, or a PET scan, or electrode stimulation, or if you put electrical stimulation, that's also fine. We can't just make sure that you have used MRI and you haven't accidentally put the letters around the wrong way. And you can also have a CAT or a CT scan as well. Any of those options, there's lots of them. Just be careful when you're using those acronyms and the letters. For things that we needed to potentially control for the experiment for part E, you can have any one, because it's only 1 mark from this list, which is actually really long. So we talked about the dominant hand, making sure they're using either they both use their non-dominant hand, same sex of the students, same age of the students, same time of day or amount of sleep, because that has to link to tiredness, which obviously we know can affect reaction time. The same amount of practice, or having never practiced before, that's normally why we use our non-dominant hand, because you're not used to kind of grabbing or catching or responding with that non-dominant hand. And then most importantly, if you talked about the equipment, I would never talk about equipment as a control variable, because the equipment is normally going to stay the same. There's not going to be anything about the equipment that could affect the experiment. However, in this case, you can talk about the ruler, but only in the concept of the fact that it should be the same weight or the same mass of ruler used each time. If you just use the same ruler, that's not enough here, because we need to be able to show that we understand that the weight or the mass could affect the catching of the ruler or the falling of the ruler. So if you use different rulers that had different weights or masses, that wouldn't be fair. So can't just say same ruler. And if in doubt, I would always go with anything else on this list as things that we know can affect reaction time. That's what control variables are about. Control variables are controlling or keeping things the same that could affect the results. And the majority of the time, equipment is not going to be on that list of things that could affect your results. The calculation,
is 0.24 as the answer, and you get 2 marks for getting the correct answer. If you got the actual answer, you didn't get 0.24. If you had correct working out, as I showed in the question, but for some reason you got the last bit wrong, or you wrote something wrong down from the calculator, then you can have 1 mark. For the conclusion, we need to be clear about how we're writing this. So student A has faster reactions than student B, because we are testing reaction time, or student A caught the ruler faster than student B. So rather than saying reaction time, just say what they did faster, which was to catch the ruler faster, or to spot a pattern in the data that as the more goes they did at it, the faster their reaction time was. So the reaction time decreased with practice or with repeated tries, because that is, again, practice we know is something that can affect reaction time. So these are the options we could have for a conclusion, but it's the way you write them. You can't just say higher number or caught in less time. It's just this idea that we understand we're testing reaction time. So it's about faster or slower reactions, or catching the ruler faster or slower, or this idea that we've spotted the pattern that as they caught it more times and practiced more, their reaction time decreased. So that's only one of those, and you can get a mark for that. Just be careful how you write it. Okay, so on to question 18. Plants need to absorb mineral ions from the soil to grow. The concentration of some mineral ions was measured in some wheat plant roots and the water from the soil they were growing in. The table below shows the result. So this is about plants absorbing mineral ions from the soil. My first thought here would be about active transport, because we hopefully know that plants absorb mineral ions from the soil by active transport. That's where I think this question is going, and that's the kind of content I'm thinking is going to be necessary for this question. They're also talking about water, so osmosis a little bit as well, or we're talking about the concentration of ions in water, so it's probably not as relevant. So we've got the mineral ions, magnesium, sulfate and potassium. Then we've got the concentration in the root tissue. So that's going to be the concentration of those mineral ions inside the plant cells, in the root cells. And then we've got the concentration in the water in the soil. So that's going to be outside of the plant cells or outside of the root tissue. So this is where we're going to have to talk about our concentration gradient. What's the concentration of the minerals inside the root tissue? What's the concentration outside the root tissue? And obviously we're expecting there to be some difference here that's going to need or require active transport, because we know that the mineral ions often get absorbed by active transport in plants. So the question says, can all these ions be absorbed by diffusion? Explain your answer. So this means you're giving reasons why. So you're either saying, yes, they can all be absorbed by diffusion, so from high concentration to low concentration, or are we going to need reasons why that is not the case? So remember, in order for it to move by diffusion, it would be moving from high concentration outside of the root to low concentration inside of the root. So we need to look at the concentrations that we've got and decide whether or not they could move from outside the plant to inside the plant by diffusion, yes or no. So in magnesium, there's a higher concentration inside the plant than inside the root. In sulfate, there's a higher concentration inside the plant than inside the root. And in potassium, there is a low concentration inside the plant than inside the root. So these two, we're going from the soil to the plant. The first two, magnesium and sulfate, are going from low to high, which we know, or hopefully know, is active transport. That's not diffusion, because diffusion has to go from high to low. So out of these mineral ions, only potassium can be absorbed by diffusion. The other two cannot be. So we cannot say that all of these ions can be absorbed by diffusion. So our answer is no. And then we need to justify or give reasons for our answer by saying because the other two, in order to be absorbed from the outside to the inside, they would need to be absorbed by active transport because they're going against the concentration gradient, or they would require energy because they would have to be moved against the concentration gradient. Okay, so we're going to tick no, because obviously all of the ions can't be absorbed by diffusion, and we've said that. So now we just need to explain our answer because there's 2 marks here. So we need to say what diffusion is, and then we can say why this isn't diffusion or why they can't move by diffusion. So because diffusion is movement from high concentration to low concentration, and for the sulfate and the magnesium ions, they would have to move against the concentration gradient, so from low concentration to high concentration. And that's our explanation. So my answer is no, and my reasoning is because diffusion is movement down the concentration gradient, or you could say from high to low concentration, and the sulfate and magnesium ions would need to move against the concentration gradient to go into the root. So there we go. That is our no, and then our reasoning. Okay, so the next part of the question says, the uptake of magnesium ions by the wheat plants was measured with oxygen present and without oxygen. The graph shows their results. So the blue line is with oxygen and the orange line is without oxygen. And it was measured over time. So we've got the uptake of magnesium ions over time. So it says we need to calculate the rate of uptake of ions between 180 and 240 minutes for with and without oxygen. And we need to show our working. So calculate question, maths question, we need to show our working, and we're calculating rate. Now rate is always going to be the change in y divided by the change in x. And that just means the change in the y-axis, so the difference between one point of the line to the next point of the line on the y-axis, and the difference in x, or the change in x, between one point on the x-axis and another point. They've helpfully given us our change in x, really, because they're telling us to just focus on the 180 to 240 minutes. So we just need to work out the difference between these two numbers, and that gives us our change in x. So what was the change in time? How much time passed in our x-axis at the point we're looking at? So if we do 240 minus 180 minutes, we get 60 minutes. So our change in x for both with and without oxygen will be 60 minutes on the bottom. So now we just need to work out the change in y for each line on the graph. To work out the change in the y-axis, we're going to need to draw lines up from the x-axis until they hit the line, and then draw them across to the y-axis to read the number. In order to do this, you will have to do it with a ruler in order to be accurate. So please, please, please make sure you have a ruler in your exam. If you don't have one, put your hand up and ask for one, and then always try and do this with a ruler and go along the lines of the graph paper if that's appropriate. So in this case, that's quite easy. For the 180 up to the without oxygen line, we're going up the 180 line and then we go nice and across on the graph paper line. For the 240, that point is in between two boxes. So we're not touching a graph paper line, we're halfway in between the two, and have to make sure that I get that right. So I'm going to move my line and make sure it's touching the point on the line before I go across to the y-axis. So then in order to do that, we literally need the difference between the two numbers where it hits the y-axis, and I'm going to need to work out exactly and know for sure what one box is equal to on this graph paper. So go down to the bottom and you've got 0 to 100 in 5 boxes. So you do 100 divided by 5, that means one box is worth 20. So now I know that my first line is 240 because it's 2 boxes above the 200. And then the line above is only halfway up. So that means it's halfway between 240 and 260, so it's 250. So now I can calculate my change in y for my line without oxygen. So it's 240 minus 250 means it's 10. And the units on this y-axis are just arbitrary units. So there isn't any units here to be used. But if you were doing this with a graph that had units on the y-axis, I would say use those units. So now I'm going to go up and do exactly the same thing for the with oxygen line. Take my lines up to those points, go across to the y-axis and read off the numbers. So my difference between my 480 and my 410 is 70. So now I can calculate my rate because we've worked out the change in y for both lines and our change in x is the same, 60 minutes for each. So I'm just going to do 10 divided by 60 and 70 divided by 60, and that will give me my rate for each line. So my rate with oxygen is 1.16 and my rate without oxygen is going to be 0.16. So part C, now using your answer to part B, calculate how many times faster the rate of magnesium ion uptake was with oxygen than without, between 180 and 240 minutes. Show your working. So how many times faster? It means, was it 3 times faster, 4 times faster, 10 times faster? That's what it's asking us to work out here. So how many times faster? All we need to do with our two numbers then is to work out how many times 0.16 fits into or goes into 1.16. It's a very simple kind of maths question that you may have had before. So we're just going to divide one by the other to work out how many times 0.16 can go into 1.16. And we're going to show our workings. We're going to write that down in our answer section, not just do it in our head or on the calculator. So we get the answer of 7.25. So the answer is 7.25 times or 7.3 times or even just 7 times faster. So part D, using the graph, explain the difference in uptake of magnesium ions with and without oxygen. Okay, so explaining the difference, reasons for the difference. We need to give reasons why there is a difference, to say what the difference is and give reasons why there is a difference between the uptake of magnesium ions. Now, uptake just means absorption, like taking them into the root tissue. And so we need to link that uptake to why there would be more or greater uptake with oxygen than without oxygen. We know it's faster. We can see on the graph there's a bigger increase between the blue line compared to the orange line. We know that there's more or a greater uptake or a faster rate or an increased rate of uptake with oxygen than without. We need to be able to link that to oxygen and say why. So we know from the table that we got in the first part of the question and our calculation that it's going to be happening by active transport. So we know from the magnesium ions in the table it's going from low to high concentration, so it must be being moved by active transport. So now we just have to find a link between active transport and oxygen. So think about it. Active transport requires energy. Energy comes from respiration. Respiration needs oxygen to happen if we're going to do aerobic respiration. So you put all of that together and it means that if we have more oxygen, so we've got with oxygen and without, but in that case there's more oxygen when there is oxygen in the blue line. So if we have more oxygen, then we can do more respiration, which releases more energy, which means that we can do more active transport of magnesium ions. That's it. That's 4 points. That's our 4 marks. So I can see how this could be quite hard, but none of this is new to you. You know that active transport is happening because we worked that out at the start of the question. You know that active transport requires energy. That's what makes it different. You know that energy comes from respiration, and you know that respiration needs oxygen to happen. So it's linking all of those things together in one sort of reasoned argument. So we're just going to write that. So the uptake of magnesium was greater with oxygen than without because the uptake was by active transport, which requires energy. More energy is released from aerobic respiration when oxygen is present. And you could say than anaerobic respiration when oxygen is not present if you'd like to, but I don't think that that part would be necessary. Okay, so the mark scheme for question 18. For A, we get a mark for saying no. And then we need a second mark for talking about how we're explaining why they can't be moved by diffusion. So we need to say that they can't all be moved by diffusion because that would be high to low concentration. And then the mark is for saying, why is it not diffusion? Because the concentration of magnesium and sulfate ions is higher in the soil than in the root. So you can say that. That obviously suggests that you understand that it would have to move against it. Or you can actually say kind of what we said, where to enter the roots, the magnesium and sulfate ions must go up or against the concentration gradient, or be moved from low to high concentration, for example. Anything along those lines where you've kind of clearly said that they're not going from high to low concentration, they're going the other way, then you can have that second mark. 18b, you've got the correct differences calculated from the graph. So if you've got 10 and 70, you can have 1 mark. If you've got the correct rates that you calculated, so if you've got 0.16 and 1.16, then you can have 1 mark. And then the times faster is only worth 1 mark as well. So if you've got 7.29 or 7.3, 1 mark. Obviously, if you've got 7.29 or 7.3, you can have all 3 marks. If you didn't show any working and just wrote down 7.3, you would still get 3 marks. But if you didn't get that, but you showed the other 2, you can have a mark for both of those parts of working out. And then part C, we need to say, so the uptake of magnesium ions was greater with oxygen because uptake is active by active transport, and that requires energy. And then this is mark 0.4 here, so more energy from aerobic than anaerobic. And that's the 4 marks. So there's actually lots there. If you just talked about active transport, you can get 1 mark. If you talked about the fact that just stated the pattern, so the uptake was greater with oxygen, then you can have 1 mark there. If you talked about requiring energy or active transport requiring energy, you can have 1 mark. I think the 4th mark here is probably the hardest to get, to link that idea of active transport and oxygen. You would still be able to get 3 marks if you didn't manage to get that bit. Okay, so on to question 19. A student stated that light was important for photosynthesis and that the colour of light didn't matter. They set up an experiment to test this with the question, how does the colour of light affect the rate of photosynthesis? They counted the number of bubbles released when pondweed had different colour lights shone on it. Okay, so this is a photosynthesis practical question. Hopefully you'll have done this practical or something similar to this practical, maybe not using different colours of light, but you'll have used different light intensities perhaps. And so really, it's just thinking about what do we know about practical, and we have to think about practical techniques, variables, and what things we're going to maybe control and look at. So sometimes they'll do this with practical questions. There'll be like a hypothesis or a question or a statement that they're testing to see if it's true. And this is how we do science practicals normally. There is normally a theory that we have or a question that we're posing, and we're seeing whether that is the truth or not. So we've got given quite a lot of context here, but not written out like a standard method, but we have to just figure out from the question what they did. So they measured the number of bubbles that were released, and what they changed were the different colours of light that were shone on it. So this is what I do with anything where there's a practical question, lots of text. Make sure you've kind of read through, you understand what they're trying to tell you with this information, and make some little notes like this because it's going to help us when we then have a go at the questions. So state the independent variable. So that's basically say or give, just tell me what it is. So what's the independent variable in this experiment? So if you haven't heard this before, you may have. I like to remember it by saying dairy milk ice cream. So DMIC for short. So the D represents the dependent variable, and the M reminds you that it's what you measure. And the I for ice cream represents the independent variable, and the C helps you remember that it's C for change. So the dependent variable is the thing that you measure in the experiment, and the independent variable is what you change in the experiment. Now, we've already got that from the text, and we've already written those notes down, so it's nice and easy for us. The independent variable, what they changed, is the colour of light. So to describe 2 ways the students can ensure that the results from this experiment can be compared with other similar experiments. So this is a common question, and it's a common way of asking a question that could be done in a different way. We've got 2 ways we're being asked to, but we'll get 4 marks. So that means we need to be able to state what is going to be controlled and then explain how they're going to do that. So, or describe how they would control that variable. So the reason it's about control variables is because in order to compare results with other experiments, everything else must be kept the same in order so that you're only changing the colours of light. And if everything else is kept the same, then you know nothing else interfered with the experiment, and other experiments, similar experiments, would do the same thing, so that means your results can be compared. We need to be able to say what variables they're going to control and describe how they will control them. So we're not explaining why we need to control them, because that would be saying what effect they would have on the experiment if we didn't. We're just describing how, sort of a method step, how we should control them. What must we control in this experiment? Well, for any photosynthesis experiment, control variables need to be based around factors that affect the rate of photosynthesis, because that's what we're measuring. So depending on what we're changing, and in this experiment, we're changing the colours of light, we will need to control all of the other factors that could affect the photosynthesis reaction. So light intensity, temperature, carbon dioxide concentration, and then there's some other things about the experiment that we could or should also control, like using the same plant, same type of plant, the same size of leaves, so they've got the same surface area. There's lots of other things that you should try and keep the same if you're using an organism in different experiments. Okay, so let's turn this into an answer then. So remember, we need to talk about the control factors we're going to control. We need to pick 2, and then we are going to describe how they can keep them the same. So to keep light intensity the same, the distance between the plant and the lamp should be kept the same. And you could describe that by saying use a ruler, or just measure the distance between the plant and the lamp and it should be kept the same. Then I've chosen temperature for my second one. So to keep the temperature the same, a heat sink should be placed between the lamp and the plant, e.g. a beaker of water. So these are all things that hopefully are part of the practical that you've looked at and ways to control these measures. There are other things we could say. Obviously, we've got the rest of our list here, and we'll look at some of the other things we could say in the mark scheme. Okay, so part C. The results that the student obtained are below. So we've got a results table where they've got the colours of light that they used. They've obviously repeated the test 3 times, and they measured the number of bubbles released in 3 minutes. And then we have a mean. Comment on the error that the student has made when recording the data. So we need to look at the data, and we need to think about the fact that they've told us that there's errors in the table that they've made. And so we need to be looking for that and then basically say what they've done and why that's a bad thing. So my main thing, and this is me, hopefully, and you as scientists looking at this table, is something's not quite right. And it's because they've got different decimal places. Some of them have decimal places and some of them don't. And that's not normal. We normally should have columns, at least in the table, should all have the same decimal places. So let's think, should it be whole numbers or should it be decimal places? So the mean is different because you weren't counting the mean. The mean can have decimal places, but they should all be the same, and they're not. But also, if we're thinking about what they're measuring in tests 1, 2, and 3, they're counting bubbles. Can you count 0.5 of a bubble? No, you cannot. So if you can't count half a bubble, you shouldn't have a 0.5 as a number, because that doesn't make sense. So we need to be able to say that obviously the decimal places are inconsistent, which isn't good. And so the number of decimal places should be the same. And you can comment on the fact that they should be whole numbers because you can't have half of a bubble. Okay, so the next question says, complete the table by calculating the mean value for green light. So using the results in the table, explain what you can conclude about the relationship between the colour of light and the rate of photosynthesis. This sounds like a straightforward question, but there's a lot in here. So using the results in the table means you need to look at the patterns of the data in the table. Sometimes this will need us to mention data, and we could mention numbers in our answer here, but it wants us to focus on the table and not give a reason based on just what we think might be the answer. You need to explain, so we need to give reasons why for our answer that we're going to say. So it wants us to look at the relationship. Relationship is just a code word for pattern. So it's normally talking about how one variable is affecting the other. So how is the colour of light affecting the rate of photosynthesis? Now, we didn't measure rate. So in our table, we don't have a value for rate. But remember, rate is just something happening or a change over time. So we measured the number of bubbles and the amount of time because it was in 3 minutes. So more bubbles means more oxygen was produced, obviously from photosynthesis. And if that's all happening in the same amount of time, you could divide it by time to get bubbles per minute, and that would be rate. So the highest number of bubbles in the same amount of time in 3 minutes was with white light, and the lowest was with green light. If you're thinking about the colours of light, though, the separate colours, then red has the highest out of the individual colours of light. So we can use all of this information to write an answer. We're going to choose what we're going to talk about. You could talk about the red versus green and talk about the colours, individual colours of light and that pattern. Or you could just say that white light produced the highest amount of oxygen in the same time. So that means it has the highest rate of photosynthesis. So we need to say what the effect of the coloured light on the rate. So white light produced the highest rate of photosynthesis because we need our reasons why we know that from the table. More bubbles were produced in the same amount of time or per minute. So that meant that there was a faster rate. So this is what we mean by explaining. So stating the pattern isn't enough here. So saying white light had the highest rate of photosynthesis, you need to be able to explain why that is the case, why you know that from the table and the data that you've got. So the rate of photosynthesis is highest with white light because more bubbles of oxygen were produced in the same amount of time. or you could put in 3 minutes, or you could put you could say per minute because you could divide it, you could work that out and show the numbers. You could say highest, you could say it's 51.16 compared to the other numbers if you wanted to. But as long as you said the pattern and then you've explained it by explaining that you understand the data about the bubbles and the time, meaning that is the equivalent of rate. Okay, the next part of the question says the graph below shows how temperature can affect the rate of photosynthesis. So we've got a fairly complex graph, but you might have seen something that looks like this before. So we've got carbon dioxide uptake in arbitrary units on the y-axis. That just means how much carbon dioxide is being taken into the plant. And obviously we should know hopefully that plants take up or absorb carbon dioxide through their leaves for photosynthesis. So the more carbon dioxide they're absorbing, the more photosynthesis they're doing. So that makes sense. It's kind of a measure of photosynthesis. So then we have two parts to this line, and hopefully we've seen these kind of graphs before. So at C, the light intensity is the limiting factor here, or as light intensity is increasing, then the rate of photosynthesis is increasing because obviously more carbon dioxide is being taken up, so that must mean more photosynthesis is happening. At D, the light intensity is still increasing on the x-axis, but the carbon dioxide uptake has really slowed down and we're not doing as much photosynthesis as before, so something else must be stopping it or limiting the rate of photosynthesis there. And then we've got some letters A and B over on the left-hand side, and we've got like a 0 line, and then the B is pointing below that 0 line. So that means below 0, or carbon dioxide is not actually being absorbed, it's being released instead. That would be the opposite. Or we're kind of a negative uptake, so it just means we're not taking up any carbon dioxide, but it's actually below 0, so it means we're putting out carbon dioxide instead. And so the putting out of carbon dioxide happens through respiration. As light intensity increases, the rate of photosynthesis increases. Well, we said that was C, and that makes sense because as you go along the x-axis, increasing light intensity, the line on C is going up, and we're increasing the rate of photosynthesis as well. As light intensity increases, the rate of photosynthesis slows down, makes the most sense for D, because we said it slows down. We're not still going up and up and up with that line like we are at C. It's kind of curved and flatlined and slowed down, even though the light intensity is still increasing. So that makes sense. So now we've got two of the other boxes left. One says the rate of respiration is greater than the rate of photosynthesis. That would mean that we were producing more carbon dioxide than we were absorbing, because respiration produces carbon dioxide and photosynthesis takes it up or uses it up. And then the other one is the opposite. So the rate of photosynthesis is greater than the rate of respiration. So that would say more carbon dioxide is being used up than is being given out. So this is going to relate to our A and B, because we've used our C and D already. So A is above that 0. More carbon dioxide is being taken up because you can see as the line goes up, or above that line, we're going up in terms of how much carbon dioxide is being absorbed. So that must be the one where the rate of photosynthesis is greater than the rate of respiration. And that makes sense because we said at B, we've got less than 0 carbon dioxide being absorbed. That must mean carbon dioxide is being given out instead, which must mean that more respiration is happening. So that makes sense for B. This was probably a tricky one, but I definitely do this with multiple choice. Go through process of elimination. Find some of the boxes on there that were easier to do first, like we did with the light intensity. Make sure that they make sense, and then use kind of a elimination process to make sure you really have looked at and understand the graph before you attempt questions like this. So, part D, explain what is meant by the compensation point. Explain what is meant by as a phrase for a question is often about defining a term, so in this case compensation point, and in this case how to identify it, or with detail enough that you could explain exactly what this is to the people who didn't know what this is. So our compensation point is represented by that 0 point on the graph. So the graph is a bit of a clue here to what they want for the answer to this question. The compensation point is the point at which the rate of respiration and the rate of photosynthesis are equal. So that means we don't have any output of carbon dioxide either way. So we don't have more uptake of carbon dioxide and we don't have more output of carbon dioxide. It's 0 because it's balanced. It's effectively neutral, 0. So that's how you can identify it. On a graph like this, that's what the compensation point is. It's where there is a balance between the two. So it's 0 output of either uptake of carbon dioxide or output of oxygen, for example, and they're about the same. So there's no overall uptake or output of carbon dioxide. On to part D. Most plants have a maximum temperature that they can still carry out photosynthesis. Explain why. Okay, so we need reasons why some plants can't carry out photosynthesis above a certain temperature. So why would they stop, or why can't they photosynthesize above a maximum temperature? Hopefully we've seen some of these temperature and photosynthesis graphs. So when you have temperature on the x-axis instead of light intensity, and you have rate of photosynthesis on the y-axis, it looks like this classic kind of hump bell curve shape. And so we know, hopefully, that most or nearly all enzyme controlled reactions have a graph that look like this, because we have an optimum temperature, and beyond that optimum temperature, the rate of any reaction that's controlled by enzymes will decrease because enzymes are denatured after their optimum temperature. So that's all we're going to say here. So after their optimal or a maximum temperature, the enzymes that control photosynthesis or allow photosynthesis to happen will be denatured, and therefore the rate of photosynthesis will slow down and eventually stop. Okay, so the mark scheme for question 19. There are 4 marks for that first question. So we get 2 marks for the control variables and 2 marks for linked measure. So you only get the marks for the how you're going to do the controlling if it's linked to the correct control variable. So you can get a mark for light intensity and you can get a mark for temperature, but you're only going to get the marks for the measurements or the how you're going to control those if you've put them with the right factor. So keeping the light the same distance from the plant for light intensity. No change in heating or cooling, or do not heat experiment. There's other things that you could do here. So carry out at room temperature, or you could talk about using a water bath to maintain temperature. As long as you've made it clear that there should be no change in the temperature and everything should be done at the same temperature, then that's what matters here for that answer for the temperature one. We talked about the size of leaves, and so therefore you're going to use the same plant for each experiment. You could talk about the same species or the same type of plant being used to kind of maintain the same rate of photosynthesis as well. Others can be allowed marks. So what else could we control here for this one? So we said about carbon dioxide concentration. So make sure they have the same carbon dioxide concentration, because obviously that can affect the rate of photosynthesis. So you would need to say they would have the same concentration or volume, but you cannot say amount. We don't use the word amount in science because it doesn't mean anything. So the same concentration or volume of sodium bicarbonate or bicarbonate of soda. So the same concentration or volume of sodium bicarbonate in each tube, or in with the pondweed, however you want to say it, every time that you're going to use it. So that's another one you could have had there as an answer for these questions. B1, you've got different resolution used, or the number of decimal places was different, which is what we said. So you can't count part of a bubble or half a bubble. You could say that they should all be whole numbers, or the number of decimal places should be the same for all of them. Any of that is acceptable. Ideally, you want to talk about whole numbers or the same number of decimal places being kind of 0, because, as we said, you can't count half of a bubble. For B2, you've got the rate of photosynthesis is highest with white light, or, as we said, you could talk about red being the highest for just using a single colour. You can say red produced the highest one out of the 3 colours. And then we need our reasoning. So our reason why that has affected the rate, and you need to link the idea of how we would calculate rate. So more bubbles of oxygen per minute. Or if you didn't say per minute, you need to say in the same amount of time, or 3 minutes, because 3 minutes was the time that was measured in. Okay, so this is where you get our second mark for kind of giving the explanation for linking it to rate. So for the matchup, we've got the answers here, just as we said. Just be clear with these. Make sure it's very obvious which line is going to which box. Use a ruler if you can. If at any point you make a mistake and you really do need to cross it out, you need to make it very, very clear that the line is crossed out. And if you're not sure, feel free to write the letters, for example, next to the descriptions if you really make a mess. But as I said, if you go through the question as we did and do a kind of process of elimination, do the easy ones first, be really sure before you draw that line, and make sure there's only one line coming out of each of the letter boxes, because if you've accidentally changed your mind and there's still a line coming out and you don't effectively cross it out, then that can stop you from getting marks. So the marks here are 3 marks if you get them all correct, 2 marks if you got 2 boxes correct, and 1 mark if you only got 1 box correct. Okay, so part D, as we said, sort of defining and describing the compensation point. So it's when the rate of photosynthesis is equal to the rate of respiration and the uptake of carbon dioxide or production of oxygen is balanced, 0, equal. And then for 19E, you've got above their optimum temperature enzymes will denature, and photosynthesis is an enzyme-controlled reaction. Doesn't have to be in that order. If you made it clear, you said something like photosynthesis is controlled by enzymes, or the photosynthesis reaction requires enzymes, and above their optimum temperature or maximum temperature, enzymes will denature.
Okay, so next we have a question about stem cells. So stem cell cloning can be used to quickly produce clones of plants. So it's about stem cells, but we're probably going to have to focus on plant stem cells at some point, and we're thinking about cloning as well. So first of all, explain the unique features of a stem cell. So give reasons why stem cells are unique, really. So what are the features that we can only describe as being found in stem cells? Hopefully you should know what stem cells do and what they are, but they basically can become any type of cell because they are undifferentiated. And we have to have that word. Try and avoid using the word specialised or not specialised, because that doesn't always get you marks. We need to say undifferentiated as a key word here, like that's definitely going to be a mark out of our 3. So there's a lot of marks on offer here. So stem cells are undifferentiated cells is going to give us 1 mark. They can divide, so they can replicate themselves as stem cells, and then they can differentiate into many different types of cell. It's the idea that they are undifferentiated, so they aren't specialised, but we need the word undifferentiated to get the marks. They can divide, so they can replicate themselves, and then they can differentiate into many different types of cell, and then obviously go on and become different types of tissues and stuff like that. So there's our detailed answer for, like, basically what is a stem cell. This would be the same answer if they said what are stem cells. That's the same question, really.
Okay, part B. Describe the key features of the following. Embryonic stem cells is the first one. Key features, just to be clear, what they mean here is kind of like unique features. So things that make these stem cells, how would you describe these stem cells and what are the features in them that make them special and what makes them embryonic stem cells or adult stem cells or plant stem cells? So for embryonic, we hopefully should know what these 3 different types of stem cells can do. So for embryonic, it's that they can differentiate into many or any type of animal cell. Adult stem cells, so this is an example of like bone, the ones that come from bone marrow, for example, so still animal stem cells, but ones that you get from adult humans, not embryos. They can differentiate into a few or some types of animal cells. And then we have plant stem cells. This one's slightly different. You can see we've got more lines because it's 2 marks. So we need to say 2 things, 2 key features about plant stem cells to get 2 marks here. Can't just say 1 like the others. So that must mean there's something extra to say. So they have the same feature as embryonic in that they can become any plant cell, but they have this added ability that they can do this throughout the life of the plant. So embryonic stem cells are only present in embryos. Then when that embryo starts growing into a child and then an adult, you don't have that ability anymore. You lose that differentiation ability. And we then just have adult stem cells instead. Whereas in a plant, the embryo of a plant can differentiate into any plant cell, and that stays the same throughout the whole life of the plant, which is how we can do things like plant cloning, like cut a bit of a plant off, put it in a pot, and it will grow into a new plant. So we need to be able to say both those things to get the 2 marks here. So they can differentiate into any plant cell, and they can do this for the whole life or throughout the life of the plant. Okay, part C, name the part of the plant where stem cells are found. Okay, this is one of those ones where it's just a fact. We have to know this fact, and it is the meristeme, which is at the tips of growing shoots and roots, but the actual kind of tissue part that contains the stem cells is called the meristeme. Okay, so part D, evaluate the uses of animal stem cells in medicine. You should arrive at a reasoned conclusion in your answer. Okay, so evaluate is always going to be about the pros and the cons. So we have to have pros and cons of both types of animal stem cells. So the types of animal stem cells we know should be adult stem cells and embryonic stem cells. And we're talking about their uses in medicine. So what can they do? What can they treat? And the pros and cons of using them for medical treatments. And because this is a 6 marker and it's an evaluate, we have to arrive at a reasoned conclusion. So you have to have an opinion, state your opinion at the end, and it has to be reasoned with some facts, some evidence. And you have to justify, say, I think this because, and give some reasons. So we're going to obviously be sort of comparing animal or adult stem cells, and embryonic stem cells. So you're probably going to have to pick which one you think should be used for medical treatments and why. And that's what we're going to try and look at. So we need to have a think about what we can remember about both adult stem cells and embryonic stem cells, the pros and cons of each, and then decide which one we think would be used and why. Okay, so we've got some pros and some cons there, which is good. So we've evaluated kind of the medical side of that bit for initially anyway. So then thinking of another pro, so embryonic stem cells is a pro because they can differentiate into many cell types, and so they can treat more diseases. So that's kind of like an alternate pro to the con of adult stem cells not being able to differentiate as much. And then they can also be sourced. So if you think about the fact that we talked about donors, they can be sourced from IVF treatments. So leftover or byproduct or waste product, for want of a better word, from IVF treatments. There'll be embryos that don't want to be used. So couples who have enough embryos, have enough children, don't want to use any more that they've kept in storage, or the couples have now grown too old, or they don't want to continue with the treatment. Any embryos that were created from IVF would then normally be destroyed, or they can be donated to be used for stem cell treatment. Okay, so let's listen to some cons then to the embryonic stem cell option. So this is where our ethical and religious element comes in. So some people view embryos as a human life, a potential human life, and they do not think they should be destroyed. Also, as well as thinking that they have this potential human life, there's also a recognition that they cannot give consent. So unlike if you were taking adult stem cells from a patient or a donor, they have consented to give their stem cells in those cases. People that believe that for ethical reasons, obviously no consent is a problem. So these human embryos that could become human lives did not consent to be used to help save someone else's life. They don't believe they should be destroyed in this process because they have the potential to become a human. And so this is often linked to religious beliefs, as well as there's obviously an ethical element to that as well. So private treatment and storing personal stem cells. So you can get embryonic stem cells from umbilical cord blood as well. But anything where you're sort of looking at private treatment to do this, to go and get stem cells privately somehow, or you've organised your own stem cell treatment from storing umbilical cord blood, it's expensive, and so this isn't available to everyone, which then becomes a bit more of a social issue, like who is able to get this treatment and who isn't. So I'm going to organise some of these points, not all of them, but some of these points into a paragraph, which would be the style of paragraph I would consider kind of putting these together. The more you can link your ideas and show your pros and cons without it being a very obvious kind of bullet point answer, sometimes the better, because these leveled answers are looking for logical, coherent, linked points. So I'm going to try and read through the paragraph, and I'm going to try and highlight the pros in green and the cons in like a dark purple. So my pros and my cons will be highlighted so that we can see where I'm putting these points into my paragraph. Okay, so stem cells can be used to treat serious health conditions and diseases such as type 1 diabetes, improving or reversing paralysis, and leukemia. This could save or improve many people's lives. So lots of pros in that paragraph. However, adult stem cells from bone marrow, which are easy to obtain from the patient with consent, are not able to treat all of these diseases, as they can only differentiate into a few types of cell. Also, if an adult stem cell donor is needed, then it can be hard to find a suitable match. Embryonic stem cells are able to differentiate into a wide range of cells. These are therefore more useful and could treat many more diseases. They can be obtained from embryos no longer needed from IVF treatment. However, some people believe that as the embryos cannot consent and they could develop into a person, that they should not be used. Some people argue that this would be destroying a life, which is against their religious beliefs. So now I'm going to end with my justified opinion. Overall, I think that embryonic stem cells should continue to be used for medical treatment and research because they can differentiate into more cell types and can therefore help more patients with different disorders. Okay, so you can see I've got pros and I've got cons, mostly in both paragraphs. They're linked together. So I've got my however statements so that I can sort of talk about something and say however, and then go into the cons to link those points together. So I've said some pros and then I'll say however to link into some cons that's linked to it. I've talked about both adult and embryonic stem cells. I've talked about the kind of medical uses. I've talked about those ethical and religious issues with consent and then the idea of getting rid of lives. I've talked about the fact that it's hard to find donors, which is obviously an issue with the medical issues. There's lots more you can talk about with this. So we could have talked about, say, the idea of rejection. So if you use a patient's stem cells from their own body, like adult stem cells, then they're less likely to be rejected than if you got it from an embryo that wasn't related to them. We didn't mention the cost thing and the talk about the umbilical cord blood, that you could mention as well. You don't have to mention all of these to get these 6 marks. It's about the range that you cover has got to be enough and that you've got a logical, coherent, linked answer, and most importantly, that we have that justified, so a reason why with an opinion at the end as well. And that will get us into that top range of marks. Okay, we can have a look at the mark schemes and see what I mean by the leveled kind of descriptors there.
Okay, so here's the mark scheme for question 20. So we've got those 3 points for A, where you've got they can divide multiple times or they can keep dividing. They're undifferentiated. That is a key word. You have to have that word to get the mark. You can't have unspecialized because that's not the same thing. Undifferentiated cells and specialized cells are different. So you can see that we've got specialized cells here. So you need the word undifferentiated to get the mark for point 2, and that they can become different specialized cells. Or I would accept here, or you probably get accept here, types of cells, because that suggests that they are specialized in some way. They're different types of cells. They're not all the same. So just make sure your language is on point for part A. You've got any type, and I also said you can have many, that's fine. Only some, you can also have few here for the adult stem cells. And then, as we said, any type or many types of plant cells. Plant stem cells can differentiate through the whole life of the plant. So something about they can do it for the whole lifetime, or they never lose that ability, something like that for mark 2. And then we've just got meristem for part C.
Okay, so the mark scheme for D, we have a very long list of what we call indicative content. So it doesn't mean that you have to have all these points in order to get the marks, because you'll see on the right-hand side, it's a leveled answer. So you need to have used this content in your answer, not necessarily all of it, but to get 1 to 2 marks, you'll only have talked about basic concerns of adult or embryonic without using specialist terminology. So things like differentiation is a special terminology word here. So talking about how they can differentiate, talking about bone marrow as somewhere we can find them, talking about a donor of stem cells, talking about IVF, talking about anti-rejection, talking about diabetes or one of these other diseases that we can treat with the stem cells. All of these are kind of classed as kind of specialist terminology. So if you only mentioned 1 or 2 things about maybe one, so just adult or just embryonic,
without talking or using some of these words, then you'll be in the 1 to 2 mark bracket. If you've talked about both types, so you've talked about both adult and embryonic stem cells, but you didn't necessarily use a specialist terminology, but you talked about one of them in more detail than the other, so you mentioned a lot about embryonic with some of these specialist terms, but you didn't mention a lot about adult, then you can still be in the 3 to 4 mark category. To get into the 5 to 6 mark bracket, you're going to address multiple areas, so both embryonic and adult stem cells in detail, so multiple points for each one, so multiple pros and cons in each one, using specialist terminology, and you need your opinion with a justified reason. That's the only way you can get into this high mark bracket, and our answer would fall into this 5 to 6 mark brackets, probably get full 6 marks. That was the idea behind me writing that model answer. So if you didn't write as many detailed points and have it split evenly between adult and embryonic, and do both pros and cons of each and an opinion with a justified reason, you can't get into that 6 mark bracket. This is what these leveled long answer questions are about. Okay, so have a read, see whether you think you're in either of those groups. Doesn't matter if you can't decide on 3 or 4 or 1 or 2 for now, but decide whether you are definitely have mentioned both. Have you used some of these specialist terms that I've highlighted? Did you talk about both types? Did you talk about both in detail or just one in detail? And then think about where you think you fall on this scale.
Okay, so on to question 21. This question is about water movement in plants. Part A says, describe how water moves from the roots to the leaves in a plant. So describe how questions are normally about steps in a process. So this is going to be about steps in the process of transpiration. So sometimes this is dressed up where it asks you to describe the transpiration stream. It might use that word. We're going to need to use that word. And it's just asking you, how does water travel inside the plant? Okay, so I've got a little diagram to remind us what transpiration is. So water gets absorbed by the roots. That's a given. But it's asking us how it gets from the roots once it's inside to the leaves. So water travels through the plant here. That's not particularly helpful. How does it travel through the plant? Well, it travels in the xylem. And how does it travel? So the reason water moves up through the plant is because of this. So because water is evaporating from the surface of the leaf, which is known as transpiration, it pulls the water up the xylem. So because water is leaving the leaves and evaporating out of the stomata, which is transpiration, we need more water to come up to replace it. So it pulls the water up through the plant. So we have this idea of a transpiration pull or a stream of water, a constant stream coming from the roots up through the xylem and then out through the leaves. And that is what the transpiration stream is known as, this unbroken kind of column of water. Think like sucking on a straw. When you suck at the top of the straw, the water gets pulled up the straw and into your mouth. And you've created that kind of suction in order to pull that up. And if you think about how tall a tree is and goes against gravity, that's what we need. So we need to describe this idea of transpiration pulling the water up through the xylem to get our 2 marks. So there's kind of a couple of ways you can say this. We need to say something about water being pulled up, and we need to say what it travels through. So water is pulled up from the roots in the xylem by transpiration. Or you can say the transpiration stream, which is this constant flowing of water up and out of the leaves. The transpiration stream pulls the water up through the xylem. Either of those ways is fine, as long as you've got the xylem in there, and this idea of pulling and the word transpiration should be in there. Okay, so now we get into the actual experiment proper. So some students investigated the effect of different conditions on water loss from leaves. So already I'm thinking different conditions means different conditions that are going to affect transpiration, because we're looking at water loss, and obviously water leaves the plant by transpiration. So I'm thinking of the factors and things that can affect the rate of transpiration. And then we've got their apparatus. So we've got 3 different setups. So we've got 3 conical flasks on 3 different balances. We've got A that just has the balance and the flask with the cotton wool with some water in it, but that's clearly the control. There's no plant there. We're clearly using balances here, which means that we must be thinking about measuring mass, and therefore we're kind of be measuring the mass that's going to be lost from the water. And then we've got our setup B has a shoot, which means that transpiration is going to be occurring there. So obviously I'm expecting mass to decrease because water is going to be being lost from those leaves. And then we've got balance C, which has the plastic bag sealed around the shoot and sealed at the top of the conical flask. So that means we're creating high humidity there. So we're going to expect that transpiration is going to decrease in that setup of C compared to B. So the actual question part B says, explain why it was important that the plant shoot had the same number of leaves in each flask. So explain why, we need to give reasons why it was important. And in that case here, it means to the experiment. So why was it important, or why was it important for what we're measuring in this experiment that the plant shoots had the same number of leaves? And we have to be able to link this idea of having the same number of leaves to transpiration. So why is it important? Based on what we're measuring, we have the same number of leaves, because how could that affect transpiration? So let's have a think about what that reason is then. So clearly, or we should understand that more leaves or bigger leaves, so we could have larger leaves, this could be what the question could be about, but more or bigger leaves would mean there would be more surface area for water to evaporate from, because the leaf is where water is evaporating, and obviously the water is leaving through the stomata. So if you've got bigger leaves or you've got more leaves, you're going to have a greater surface area with more stomata, so that the plant's going to be able to lose more water in the same amount of time. So that would be unfair, especially if we're trying to compare the difference in transpiration rate or the amount of water that's being lost between B and C. Then we can't have different numbers of leaves or different numbers of stomata. Obviously, having the same number of leaves from the same type of plant doesn't necessarily guarantee that we're going to have the same number of stomata, but it's much closer than if we had different sizes of plant shoot. So that's all we need to say. So we need to explain why. So why would it be unfair if they had different numbers of leaves? Link it to transpiration, because if they had more leaves or bigger leaves, then there would be more surface area for water to evaporate from. So the same number of leaves means that the shoots have the same surface area or the same number of stomata. Either of those is correct here. You can say both as well, but either or is fine. So the same number of leaves means the shoots have the same surface area or the same number of stomata. So the same amount of water can evaporate or be lost from the leaves. And that's where the 2 marks come from here. So 1 mark for saying the same number of leaves means the same surface area or the same number of stomata. That's 1 mark. And then the second mark comes from saying so the same amount of water would be lost. So part C, apart from the size of the plant shoots, so not mentioning that, state 2 other conditions that the students should have controlled in the room where they carried out their investigation. So we're not talking about plant shoots here, we need to give 2 other conditions, even though we're only going to get 1 mark here, but that's because it's quite an easy question. It's just, can you recall some conditions that affect transpiration? So we need to know the conditions that affect transpiration. We've already talked about some of them. And we have to think about which other of these conditions are they going to control in this experiment, i.e. keep the same so as not to affect the experiment. So that really means that we have to understand here what is the factor they're changing. So clearly, we've mentioned this before, it's humidity. So that plastic bag is looking at the effect of humidity. So if we're talking about humidity, and humidity here is what we've changed, it's our independent variable, then we're not controlling that. We're not controlling humidity. So we need to think of some other factors that affect transpiration that we need to keep the same here. So if we can't remember, those are temperature, light intensity, and air movement. So often the other examples you'll see of these types of questions that they've increased the temperature or they've used a fan to increase air movement. So we need to keep those 3 things the same. Now I would go for temperature and light intensity because they're easy and they're easier to talk about, easier to remember as well. Air movement is sort of harder to say how they would control it. Just make sure the windows and doors are closed so there's no drafts. That bag around the plastic bag is going to help control air movement as well. So I would say temperature and light intensity are my go-to. If you put air movement though, or wind of some description, then I think that you would also get the mark for that. So part D, wants us to explain the difference in the mass of water loss between balance A and balance B. So we've got our table here and we've got the time in hours. So time at the start was 0, and then we're looking at balance A and balance B. We're not looking at balance C here. That X and the gap, that's going to be something for another question probably. Here we're just focusing on balance A and balance B, and it wants to explain the difference in the water that's been lost. So obviously, because we've measured the mass in the first after an hour and then after 2 hours, we're thinking about the fact that balance A, if you look at it, the mass has stayed the same. Hasn't changed entirely down the column. So even after 2 hours, the mass of balance A is still the same. Whereas the mass of balance B has clearly decreased. So you can see down this table that the numbers have decreased. So the balance is showing that the beaker and the plant chute has got lighter or has lost mass. So explain always means the same thing. We need reasons why. So we need reasons to explain why balance B lost mass, but balance A did not change or lose mass. So the mass of water lost increased, or the fact that water was lost, when a plant chute was added, because clearly it's the water that we're losing. This question is about water, and we should hopefully know that there is a way that the water can be going up through the plant and leaving the chute, and that method is transpiration. So our reasoning for why balance B is losing water is because it has a plant chute, whereas balance A does not. And therefore the water is able to travel up the stem of the chute and leave the leaves via transpiration, so evaporation of water through the stomata. And that means that water is able to leave. And so we are losing water through the leaves of the chute. It can't do that, now. There is no plant in order to facilitate this movement of water.
Okay, so the mass on balance B decreased or was less than balance A, or balance A didn't change but balance B decreased, or the mass on balance B decreased. As long as you said it was lighter, it was less, something along those lines, because water was taken into the plant chute or into the stem of the plant chute or pulled up the chute stem, anything like that, and then the water evaporated from the leaves. So we have to say all of those things. We have to say what the difference is. So the mass of balance B was lower or it decreased, whatever you need to say. That's the difference. That gets us 1 mark. Because the water was taken up into the stem, or water went into the stem, that gets you another mark, but that isn't the final explanation of why water was lost, because remember, we're weighing the beaker and the stem, so it's not going to give us a decrease in mass if water just went into the stem and the leaves of the plant. We have to say for the final point, to get the 3rd mark, it evaporated through the leaves, or it was lost through transpiration, or it left through the stomata of the leaves. It's something where you're giving a very clear impression that the water was lost through the leaf. It left the plant and therefore is not included in that mass, and that's why that mass decreased.
Okay, so part E. What mass of water loss would you expect to see at X in the table for balance C after 2 hours? Explain your answer. So because we're explaining our answer, we have to give reasons why. And what this type of question is asking you to predict the results or predict the outcome of an experiment. So what would we expect to see at X in the table? So we need to predict a number, a mass. So we've got to work out what number is going to be the most appropriate number, or a range that this number needs to sit in. It's not going to be just one number and that's the only number you can get to get the marks. It will be within a range, and we have to use the information we've been given in the question so far in order to work that out. And so we need to say what's going to happen. So is it going to increase or decrease? That's our first mark. And then we're going to need to say why it's going to happen. So increase or decrease with our prediction of a number, and then the explanation as to why we went for that number. In order to focus on the answer for C, we need to compare it to bag B in order to get our range.
So in B, water is freely evaporating and leaving the leaves because of transpiration. That's also happening in C, but all of that water that's evaporating, that water vapour that's then being produced, is being trapped inside that bag. So the water is going to build up, the water vapour is going to build up, and it probably will condense on the bag as well. So you'll see water droplets settling on the bag, and that air will be very humid in there.
And so that's going to mean that transpiration is going to decrease. Humidity increasing is one of the only factors that actually causes transpiration to decrease. All of the other factors increasing normally increase transpiration, but humidity increasing decreases transpiration because there's less of a concentration gradient between the air and the amount of water inside the leaf, so therefore it's less likely to evaporate. So it's important to note that in the table it's just the mass of the shoot and the beaker, so we know the bag isn't going to be included in the final mass. So we know the mass can't be no change, it can't be the same as balance A, because some water is still being lost and some is going to be lost and be on the bag and in and around the bag, because transpiration is occurring because there's a shoot in plant C when there isn't in plant A. We know that balance B, it's going to have to be less than this number here. So this mass at 148.1 is going to have to be less than this because it's lost more, it's going to lost less water because transpiration is going to be less, or going to be happening slower or not as much compared to balance B because there's no bag there. So we need a number that's going to be less than 150 because it can't be the same because the 150.01 is what we started with, but it would need to be more than 148.1. Because 148.1 would mean that it lost the same amount of water as balance B, and it would have lost less because there'd be less transpiration. So any number in that range would give you the right answer here. And then our explanation is obviously going to be based around the fact that we're having a bag that's making it more humid, we're losing less water. So I predict the mass will be 149.7. It could ask you to put that in the box. You could have put it in the box. You can just write 149.7 because, but make sure you've got the grams on there because that's the units. But that's what I'm going for. I've picked my number that's in between 150 and 148.1. This is because the bag increases humidity around the leaves of the shoot, so less water will be lost through transpiration. In reality, adding the part about less water being lost through transpiration is kind of an extra because we've clearly already shown we understand that water is going to be lost and less water is being lost than balance B, which is the point of this question. But we've given our reasoning and I'd like to follow through with the explanation just so you're clear but we're all clear about what we mean, and the examiner will also be clear about what you mean and that you understand why you've chosen that number. You didn't just guess it at random. Okay, so the last part of the question. So this is one of those sort of long answer questions about some data, but realistically it's a xerophyte or sort of desert adaptations question, but it's all linked to transpiration. So scientists have collected data to compare two plants, pansies that are grown in gardens in the UK and agave plants that are a type of cactus that's adapted to live in hot, dry deserts. Use the data they found in the table below to explain how the agave is adapted to live in desert conditions for 4 marks. We've got two plants, we've got pansies, so they are obviously growing, it tells you they grow in the UK, so they're used to quite cold, quite wet environments. And then it wants us to use the data that's been given. So we're sort of comparing the agave, which lives in these specialised conditions and that will have the adaptations, to a sort of a normal plant, which is the pansy. And it wants us to only talk about the differences in the table. So we're going to have to talk about what the differences are or what the agave has that's different to the pansy, and then give reasons are an adaptation to living in desert conditions. So give reasons why it can live in hot and dry environments. And it's all going to link to either reducing transpiration or reducing water loss, or storing water or getting access to more water. That's the whole point of these kind of xerophyte adaptations, the desert kind of cactus plants. It's all about how can we conserve water or how can we prevent us losing too much water, which is obviously linked to transpiration. So the first one is the thickness of the waxy cuticle. So the agave plant obviously has a thicker waxy cuticle, so we need to say there's thicker cuticle or more cuticle, and so therefore it's going to be more waterproof, so less water is lost. This is just, you know, what the cuticle does, and if you've got a thicker one or a larger one, how's that going to be an adaptation, or what's that going to create? The total leaf surface area, so you've got 1,800 versus 150 here. So the agave has a smaller surface area of the leaf, or the total surface area of the leaves is smaller, so therefore we're going to have less water loss. For the same reason we talked about with our shoots and why the plant leaves need to be the same size. Bigger leaves, larger area, the more surface area you have for water to evaporate from. Water storage tissue in the stem as a percentage, so 50% of the tissue in the stem of a pansy can store water, whereas 85% of the tissue in the stem of an agave can store water. So if you've got more tissue that is able to store water, then you're going to hold on to water when there isn't enough. So you can absorb a load more water when there is a lot of water, so when it rains, and then you can store it to use when there is less water. So it allows them to survive things like drought or survive periods where there isn't enough rain or enough water for long periods of time. Having fewer stomata, again, we talked about this with the leaf surface area in our shoot in the experiment. So if you have fewer stomata, you're going to be able to lose less water through transpiration because there's simply just fewer holes for it to evaporate out of, and so it will slow down that rate of transpiration. When are the most of their stomata going to be open? So in the pansy, it's normally the daytime, which is what most plants do. They need to do photosynthesis, they need to do gas exchange, and also that they will obviously then lose water during the day. But the agave plants, they open most of their stomata at night. So that means, or why would they do that? What's the reason? It's cooler at night time in the desert, so it's going to reduce transpiration that they would have lost in the heat of the daytime. So during the day it's too hot. If they open their stomata, they'll lose loads of water, so they're going to open them in nighttime instead. And then finally, the horizontal spread of their roots. So how far sort of away from the plant, area around the plant, do the roots spread out from? So they only spread out about 0.2 meters in the pansy. They mostly go straight down, but with the agave, they're spreading out 5 meters from the plant, which is a really large area then, so a large flat area away from the plant, and they'll be close to the surface. It just means they can cover a large area and they can therefore get access to absorb more water. You can also say it helps them absorb water really quickly after a rainfall, because if they're shallow roots that are spread really far away around where all the rain is hitting the earth, then they'll be able to absorb it faster. Either of those is fine. So for these, we've obviously said the difference. I've not done it for all of them. So for water storage tissue, I didn't say specifically that there was more storage tissue. I should have done. And for when the stomata are open, I should have said that they're opening them in the nighttime, not the daytime. That's the difference. So we've stated the difference, that more cuticle, smaller surface area, more storage tissue, fewer stomata, opening at the night, not the day, and a larger area. That has to be part of our explanation. So they've given us the table, they've not given us the adaptations. We've had to work out what the adaptation is, the feature is different compared to a normal plant, for want of a better word, and not a desert adaptive plant, in order for us to figure this out. So you would need to talk about the feature difference, or the adaptation, plus the explanation to get 1 mark. And this is a 4-mark answer, so we're going to need to pick 4 things to talk about. And we need to make sure we say the agave have more, or they have smaller, or they have fewer, or they have larger as part of our answer, and then the explanation, because that's what it wants here. It's one of those ones that doesn't look like it's a long, really super long answer question. The language has to be right for you to get marks. If you just said the agave can store more water and the agave will lose less water, or the agave can get more water from its roots, but didn't specifically say the feature, you wouldn't get the marks. So I'm going to combine 2 and 1 for the first point. So the agave have smaller leaf surface area and fewer stomata to reduce water loss through transpiration. And I've done that because they both have the same, sort of the adaptation part of that is the same. So my third point technically is the agave had more widespread roots, so they can access a larger area to absorb water from. You can also just say here they can absorb more water. And finally, the agave open their stomata at night when it is cooler to avoid the warmest times of the day and reduce water loss. Probably could have worded that slightly better and said the agave open their stomata at nighttime instead of the daytime, because it's cooler to avoid the warmest times of the day and reduce water loss. That's it. This is one of those ones, there's lots of parts to this question, most of it about a practical. Again, a practical you won't necessarily have done because it's not required, but one you're expected to be able to interpret and understand. We've had some predict questions, which are always just use your knowledge to try and predict an outcome of an experiment. Some of the hardest questions that are found in GCSE. And we've done some thinking about transpiration, its effects, and factors that can affect the rate of transpiration as well. So hopefully that was helpful.
So here's the mark scheme for question 21. For A, you've got transpiration stream or pull. Either of those words is fine there, and you need to say the word xylem to get the second mark. For B, you've got same surface area or
same number of stomata, because they equate to the same thing. Or you could say if they use a different size of shoot, there could be a larger or smaller surface area. So just linking the idea of a different leaf or different number of leaves, or different larger or smaller surface area of the leaves. And then you need to link that idea that if you've got a larger surface area or more stomata, or just a different surface area or a different number of stomata, then you've got the same amount of water, or the water needs to evaporate from there. And you can say that a bit easier by saying either more or less water will escape from those stomata or that leaf surface area. Any one from temperature, airflow, or light intensity, because all of these will affect the rate of transpiration for part C. For D, the mass got less, or you can say it decreased. You can say it got lighter because water was taken into the plant, or the roots of the plant absorbed water, and then they lost it through transpiration. Key word there. But you can say they lost the water through the leaves, or the water evaporated from the leaves, because that's the same as saying transpiration. And then, as we said, for our prediction, any number between these numbers is fine, because as long as it fits in between those two, the range that we said, then that is an acceptable number that gets you the first mark. And then you need your reasoning. So why is that the case? Well, the plastic bag reduces the airflow across the leaves, so it reduces the effect of wind or air movement. Or, as we said, the bag is creating a humid environment around the leaves. So you can either just describe that. So air becomes saturated with water is the same way as saying it becomes humid. Or the air stops water vapor from leaving, again, so it builds up humidity. There's lots of ways you can say that, but as long as you're basically explaining that you understand that it had to be less water lost because you're creating a humid environment. And then part F, as I said, there were lots of things we could say here. And there's only 4 marks available, but it seems like it was one of those quite long answer questions, but you just had to choose 4 things. The main thing is they need to be in pairs. Either you can have 1 and 2, or you can have 3 and 4, or you can have 5 and 6, or you can have 7 and 8, or you can have 9 and 10, or you can have 11 and 12. So you just need 2 pairs, and you get 1 mark for each point in your pair. But the idea is that those points should come together. You can't just say any 4 random points of this list, because obviously 1 and 2 are linked. So if you have a thicker cuticle layer or a waterproof cuticle, it keeps the water in, and that's how you're adapted to stop losing water. You have a smaller surface area, you have less water loss from your leaves. And so you need to have them in pairs, really, and then you get a point for each pair.
Ouch! This is why in some videos I explain scratches.